So I'm going to share my screen. All right, so my screen should be on. Can you see my screen?
Yes, yes, we can. Yeah.
Yes, but maybe not on the presentation, on the website. Not sure it's working on the website. Is it? Well, I guess. So I'm Encarnita Mariotti-Ferrandiz. So I'm working in Paris at Sorbonne University. And today I'm going to introduce our work in the lab about identifying TCRs as biomarkers of autoimmune diseases, which is linked to the previous talk. So what can we learn from the TCR repertoire in autoimmune disease? So I'm not going to enter into the description of what is a TCR repertoire, just to emphasize that it's a collection of antigens, specific T cells, rearrangement, and that each T cell expresses a unique TCR alpha beta, which is generated by random somatic recombination and selected in the thymus, but also eventually in the periphery upon immune responses. And it has been estimated that for in humans at a given time point, there's about 10 to the 8 unique TCRs per individual. In autoimmune disease, there have been many studies trying to decipher the repertoire of patients compared to healthy volunteers. And that has been mainly done in type 1 diabetes, systemic lupus erythematosus, and rheumatoid arthritis. In all those studies, the conclusion was that the patient repertoires were different from healthy volunteers at different levels of either diversity or composition. However, most of the studies were done on global T cells without looking at the different subsets. And you all know that autoimmune diseases are, for many of them, not all, but for many of them, due to imbalance of between T regulatory T cells and effector T cells within the CD4 compartment. So we believe in the lab, and this is one thing that we are really trying to focus on, is that the TCR in different subsets might be crucial to make any link with the pathophysiology. So this is what we try to address by studying a cohort of autoimmune patient as well as control disease, which is mainly a patient with inflammatory diseases, and healthy volunteers within the transimmunome trial. And our goal was in a first step to try to determine whether the TCRs could be a biomarkers of those different autoimmune diseases. So for that, we perform airr-seq TCR alpha beta sequencing on blood CD4 T effector cells and CD4 regulatory T cells that were sorted, and for which we applied molecular biology and next generation sequencing. The idea behind was to try to see whether we could identify disease-related TCR signatures, and what we call a TCR signature is a collection of TCR that could define a condition compared to another one. So as you can see in this slide, we had at least 100 patients expected for some of the group, at the minimum of 50 patients. So we really needed to have robust and reliable methods to be able to compare all those data, especially because it was a trial that lasts for several years. So the collection of blood and the preparation of the samples would have been probably, and which has been the case, delayed over time. So something that it's obvious now is that there's many methods to perform air sequencing that have been developed either by academic labs or by different companies. And those methods differs by different points. One is the nature of the template that it's used, either RNA or genomic DNA, which can make a big differences in terms of output. The difference is also it's relates to the amplification methods behind the molecular biology protocol. So most of those different methods use either race PCR, which is using a universal primers, and others, most of the gDNA-based methods are based on multiplex PCRs, which means the presence of the different primers for each V segment and J segment, sometimes the C segment, in the same reaction. And the third important point is the application type of samples that can be handled with the different methods. So either some of them have been developed and are mainly used on bulk cells, meaning all the cells pulled together. And more recently, some providers and some labs developed single cell approaches. So in our case, it was really important to find the methods that was the most appropriate, and we aimed at looking at bulk repertoires because we were looking at the blood on a small volume of blood. So to identify these methods, we launched international collaboration in order to benchmark the several methods that were available. So the protocol that we used was to start from human T effector cells that were polyclonal, and for which we extracted both RNA and genomic DNA to generate several aliquots. We also use a JORCAT clone. I'm not going to enter into the detail for sake of time, but we use this as an internal control for the calibration, for the control of the different methods. What was done was that for each method that was analyzed, we had 3 aliquots per dilution and 3 levels of dilutions of the JORCAT clone. So at the end, we had 9 aliquots that were analyzed by 9 different methods and sequenced in a similar manner. I will not detail all the results of this study because it has been published recently in Nature Biotechnology. But what I wanted to raise is that by comparing all those methods that were, for most of the methods, highly used and published, we found that depending on the TCR alpha or TCR beta chain, the results are different. So the methods are not as performant on both chains. Here, what I'm representing is each method on the different colors, and the similarity between pairs of samples and aliquots. For each method, there's 9 aliquots, and then we compare the different, the similarity in terms of clonotype composition. As you can see, for TCR alpha, there's 2 methods that are quite similar. The redder is the dot, the most similarities. For the TCR beta, it's a bit more large. There are more methods that are quite similar between each other, with 1 method here that was one of the most similar. But that was just comparing the methods between themselves, and there were some leaks in terms of sequencing depths depending on the methods. The number of sequences that were obtained were a bit different. So we wanted to have internal control without having the ground truth of this repertoire from one individual. So what we did was to generate a control meta repertoire that was built with sequences coming out of the different methods. So basically, we removed from all the noise by pulling replicates, removing singletons that could correspond to sequencing errors depending on the method. We also removed the private clonotypes that were private to one method compared to another one to remove the weights to this method. And we also keep only clonotypes that were observed at least in 3 methods. Then just to be sure that we are comparing the same amount of information, we perform a downsampling to the smallest data set. And then we pulled all the data sets together, generating this control meta repertoire of all the TCRs, alpha and beta chains that we have been able to amplify from this unique sample. And one of the major results that we obtained was looking at the sensitivity. And we were quite interested in this point because in our case, we were analyzing, we are interested in autoimmune diseases. So as Victor mentioned, we are usually looking for very rare events. So we were really looking for methods that was sensitive enough to provide the rarest clone. And so here I'm representing the frequency of the clonotype in each methods that were present in the control. So meaning that this dot, for instance, it's a clonotype that was present at 110 minus 10 to the 2 frequency in the control. And the result was were quite obvious when we looked at the clonotypes that were present in all the replicates, all the 9 replicates per method. We could identify 3 methods, RACE3, 5, and multiplex PCR1, that were the methods the most sensitive in detecting rare clonotypes. So based on this and different analyses that we perform, we establish a comparative performance of those 9 TCR sequencing molecular methods that we compared. And we identify 1 method for which the scores of replicability, reliability, and sensitivity that defined the performance of the different methods were the lowest, meaning the best. So based on this, we've selected the RACE3 for our transimmunome trial. And on all the data set that we obtained, so currently we did not reach yet the preparation of the library for all the samples. So the result I'm providing here are preliminary results on mostly half of the data sets. We apply a feature selection and machine learning strategy to try to identify signatures of TCRs. So these methods, applying this strategy, we could identify for T effectors and regulatory T cells different signatures on 2 major diseases that were analyzed, T1D, airr-c, compared with healthy volunteers. So in that case, these heat maps showed the presence in yellow and the absence in black of the clonotypes that were selected as features and better and the best collection of TCRs separating the 2 groups compared. So also, as mentioned by Victor, in the Emerson paper, you can see that the number of CDR3s that are able to separate the different groups are usually quite small, especially when we looked at the disease T1D for T effectors or for regulatory T cells. For airr-c, it seems that we have a bit more, but still 1,000 CDR3s, it's not too much. So now we have those collection, and we wanted to see whether these different signatures that were unique to each disease compared to the healthy volunteers were similar between diseases. So, but before we tested the prediction power of this those signatures. And one thing that I did not mention is that the signature here in this slide were obtained in 75% of our data sets. So we tested the prediction of the remaining 25% of the data set of these signatures, and we compared this prediction to a random signature that was generated. This random signature is a collection of TCR with the same number of TCR as our signature and the same characteristic in terms of CDR3 length and composition. So as you can see, for T effector cells, we could, the signatures that were obtained for T1D and airr were able to predict the remaining 25% of the samples, meaning classifying properly the remaining 25% of the samples, which was not the case for the regulatory T cell signature. So still, we wanted to see whether those signatures were specific to the different diseases or unique to each disease. And when we put all together each clonotypes that were found in the different signatures, we could see that they were able to classify and to separate the different groups of individuals and immune status that we've been analyzing. So with here, the healthy volunteers, the T1D patient that were separated here from airr patient here. That was mainly true for the TF. Again, it was less true for the regulatory T cells. We tried to see whether those signatures were able to predict the different diseases, but in that case, we did not have enough samples to do the 25% validation. So we used repeat cross-validation, although it's not the best. But as you can see, for the T effector cells, we can those signatures can easily predict all the different status. Again, for the regulatory T cells, the prediction was not bad, but it was lower than for the T effector cells. So finally, for the we applied the same strategy, but this time to external data sets, which is not related to autoimmunity. But I wanted to show you this result because it was first made on different data generated with different methods, and also from data set obtained from total blood, so without sorting T cells. So we took the large data sets provided by Adaptive Biotechnologies on COVID-19 patients, and we selected only the patients that were labeled as recovered, so they recovered from the disease, compared to the patients that died after the disease. We applied the same approach of feature selection and machine learning, this time on the TCR beta, and we could find a TCR beta signature that was able to predict with a quite good accuracy the patient that recovered compared to the patient that passed away. And that was significantly higher compared to the random signature. So those results were quite interesting at different levels. First, our methods could be applied to different type of data and different types of settings. Nevertheless, because here we are looking at total T cells, we don't know what are the cells involved, and therefore it's difficult to make the biology behind those signatures. So to conclude, so from what I've been showing to you, I hope I convince you that first, the TCR sequencing methods are not equally performant. So it's important to choose and to validate the method that suits your scientific question, especially if you are looking for rare events. It's important to use methods that are able to that are sensitive enough. It's important also to have methods that are reliable and reproducible, meaning that each time that you would perform a new experiment, you are almost sure to obtain more or less the same results. The second point, I hope I convince you, is that using bulk TCR sequencing from sorted T-cell subset may increase the chance to capture rare rearrangement. And this by showing you that we identify disease-related signatures in T1D and airr. We also predict the response to the treatment in SLE patients, so that I haven't shown because of lack of time, but showing that we can also apply this kind of approach to other type of conditions. And finally, this approach was also applied to TCR-seq from PBMCs that could maybe identify major modification, but we haven't gone into the details of the signatures. And finally, I just wanted to make the link between this work and all what the work done by the airr community, because still there's a lot of things to solve and remaining challenges that the airr community is tackling. As mentioned by Lindsay, the controls and the calibrators to standardize the methods are a really important issue that the biological resource working group is trying to work on. We also need for public and standardized data. So again, we have in the community two working groups and subcommittees dedicated to this important point, because as mentioned by Victor again, for machine learning and all these approaches, we need more data. We also need new tools and more strategies for characterization of the data, like specificity inference, epitope prediction, as mentioned by Victor also. So again, we have different working groups working on this. And finally, to determine the biological role and diagnostic interest of this the airr-seq data that will be helpful, hopefully, for the clinics. And again, this the airr diagnostics and the legal and ethics working group are working on this. So with this, I want to thank all the contributors to the work, especially the lab members, David Klatzmann, who is the PI on most of the projects with who I'm working, Pierre Barren and Paul Stis, who developed the machine learning strategy. All our collaborators from the methods comparison, without who this work couldn't be achieved, and of course the facilities and the airr community and the Antibody Society for providing us the opportunity to share our work. And I thank you very much for your attention. And if you have questions, I can't see any questions. So hopefully it was interesting enough. Thank you.
It was very interesting. Oh, sorry, go ahead.
Just quickly, for the second part where you look at the autoimmune and the antigen-specific response, did you check also whether they were already sequenced in the BDJDB database or in MCPass, whether people have found them before?
Not yet. This is the next step for the characterization of the signature. Absolutely. This is something that we are doing in most of our data using a VDG database, MCPass. We are also looking at the IEDB database that contains complementary information, but definitely this is something, and usually the results are quite interesting, but again, as you mentioned, what does that mean?
Yeah, thanks. Very nice talk.
Yeah, great talk, Encarnita. Very interesting. Could you just clarify the nature of a signature? Is it like one specific CDR3 sequence, or is it a combination of multiple and all must be present in a repertoire to flag a positive label, or is it even more complex than that?
It's a collection of CDR3s. So we are not taking into account the frequency of the CDR3s, just the presence or absence of the CDR3s. It takes into account the sharing between patient and also the comparison of the CDR3 in our dataset compared to a theoretical distribution. So, but it's a collection of CDR3s.
Okay, thank you.
So, Encarnita, I put something in the chat, but I'm not sure it made it through. I was curious why you think that it might be easier to find these signatures in effector T cells than in regulatory T cells.
So that's a great question also. Actually, so first we were quite disappointed because of course our idea was to find Treg signatures in the autoimmune compartment. So one possibility I can see is that maybe in the patient we are only analyzing one time point. So in all the patients, these Tregs might not be very efficient. I mean, they are not very functional. So what we found in some mouse model is that eventually there could be a lower. So maybe if it's too private, if the Tregs are too private and they are lower in terms of functionality and maybe diversity, maybe that's that could be the reason for why we don't see we can't see signatures in those diseases. What was a bit reassuring, because I could not see why the technical bias related to the machine learning, but why not? But what was quite reassuring is that in a setting where lupus patients were treated with low-dose IL-2, which is targeting regulatory Tregs and inducing a proliferation of those Tregs, in that case, we could identify a Treg signature. So it might be because of the privacy of the regulatory T cell repertoire, which is something we are seeing in different settings.
Thanks, very interesting.
All right, so I think it's time to move to the next talk.